Cell-free protein expression for insulin production. The problem our project aims to address is restricted insulin access in low and middle-income countries. Diabetes is a life-changing condition where patients rely on external insulin to regulate their blood sugar, and without it, their health and quality of life is severely impacted. Despite the fact that more than 4 in 5 adults with diabetes live in these low and middle-income countries, they consistently face the highest barriers to access, with limited or no availability of many insulin formulations. The primary challenges with insulin access include the inconsistency of refrigerated logistics and the prohibitive capital costs associated with establishing insulin manufacturing facilities. A simplified explanation of insulin is that it's a small protein that our bodies need to metabolize the food that we eat, to help transform it into energy for our cells. People with diabetes, both type 1 and type 2, don't have enough or any insulin at all. Fortunately, we can use external insulin formulations to help regulate the blood sugar of people with diabetes, usually via injection. The solution that we're proposing is localized, small-scale manufacturing of insulin through a modern biotechnology called cell-free protein expression, which is a method for synthesizing proteins outside of the living cell. Traditional insulin manufacturing techniques require the growth and maintenance of large cell colonies, which are time, energy, and water intensive. The cell-free method permits the synthesis of insulin using purified components, which can be shipped globally without the need for cold storage. We aim to assess the feasibility of this kind of local-scale production facility for insulin injectables, using bioprocess simulation software and a complete economic analysis. We'll also make appropriate considerations for the safety of the product in accordance with EU pharmaceutical standards and all good manufacturing practices involved with pharmaceuticals. Of course, the primary anticipated benefit of our project would be improving the health and quality of life outcomes for diabetes patients in low and middle-income countries. Reliable insulin access is life-changing, and we aim to demonstrate that this technology could feasibly provide insulin for those in need. Beyond that, we plan to demonstrate through an economic analysis that localized production facilities could be constructed with modest capital investment, ideally encouraging pharmaceutical companies to invest in these regions with underdeveloped insulin access. Also, due to the nature of the cell-free protein expression method, the heavy water burden of cell growth is shifted away from these low and middle-income regions.